each row like a study.
Each row has already run.
That's what I just used.
Are you actually analyzing all of them?
I'm trying to parse it, but not for first thing. It's like
Ratio.
look at smart 6 in terms of
I guess each one has a set on the sheet states version. So deduplicated and then into actual samples. Like if you took the unique of this row.
Yeah.
Yeah, maybe this one. Yes, sir. Equals unique and then new. That's it. Now do and unique. Count unique or no? I don't know. In pounds it's count, but I don't know. And but I don't know about next time. What it means is basically that it's never going to change.
Welcome to the first AGI House Virtual Sound Challenge. Let's hear it for what everyone's about to build. Woo! So, for me, today's a glorious day. There's been a long-term sense that AI will be capable of general scientific reasoning, but we are limited. We're limited by simulation environments, and the ARC Institute has put together the first high-quality public environment for cell simulation. And so I'm stoked to be here. The speakers will be the folk who put it together, and the people who are currently on the top of the leaderboard for the virtual cell simulation. And let's be clear, this is a moment of huge potential for progress in bio, categorical progress, where the in silico development of drugs, the evaluation, whether it be of proteins and protein binding, of genomics, of, in this context, whether or not we can get, this ability to predict weather perturbation in the context of, cell, in very many different types of cells, many different contexts in which the cells are transitioning, whether we can create a reliable predictive system for those things. It was not at all obvious, I guess, as a brain, we have this brain genomics team, we have medical brain, and folk were very ambiguous about whether, Demis had us at the time, with this concept of the virtual cell, whether this vision would come to fruition, whether we'd be able to build the simulation environments that would make this kind of thing work. And it always felt like an impossible vision. So it is kind of crazy that we are here in 2025 with the ability to, in a single day, train models that are competitive in, in this case, a small perturbation example of what I expect over the years to come, in 2026 and 2027, will be higher and higher quality examples of sort of cell activity and how it changes in the face of modifications. So first of all, fantastic that this exists and fantastic that we're here. The first speaker is Ryu. He actually is now second, but when we organized it, was first on the leaderboard for the Virtual Cell Challenge. And he'll be talking about the kind of techniques and model architectures that have been successful in letting him build high-quality models for this problem. Now, the core idea behind this event is not just to learn from folk who are doing well, it's for you yourself to come up with creative ideas, whether it be new data that you can train on, new model architectures that are efficacious, new training paradigms, new algorithms that happen to be effective in this context. So that as a community, whether it be folk who are publishing in the scientific world or building companies on the backs of this new technology, so this collective can basically create a level of collective knowledge on how to train models for this kind of problem. We'll hear a bit about how to interpret these models, which are essential for coming up with the causal structures and the pathways that are leading to the kind of changes that we're seeing. But practically speaking, there's a developing skill set, which is the ability to create high quality cell simulations in the face of high quality data sets, and the ability to construct data sets that allow us to build models that are very effective. And so hopefully you can take that as a call to action to jump personally today into the arena of creating high quality cell simulations with the models that we've trained. Lots of thanks to Hyperbolic. So if you have compute already, that's fantastic. If you don't, please try to team up with someone who does so that you can put out a high quality model by the end of the evening. The high level intention is to basically kick off talks at about 1 to have people start training their models and setting up the data structures and processes such that it's possible to create a high quality training pipeline, and then to get results, ideally before the end of the evening, that we can post to the virtual cell competition. And my hope is that we can be competitive. I think with the data that you have access to, according to the winner at present, it's possible to get top 20. And so hopefully people have a lot of ambition and can place well in the competition. With that, let's hear it for the top 2 performer on the Virtual Cell Challenge, Rio.
All right, thank you so much for having me here. Okay, just a show of hands on the tech to bio spectrum. How many people would you say are on the tech side? Okay, and then how many people would you say are on the bio side? All right, okay, sounds good. And just to give you a little bit of background about myself, I would probably raise my hand about halfway for both of those sides. Okay, so before I get started, I want to give you one caveat, and that is that, as Jim mentioned, I'm number 2 on the leaderboard, and probably will be lower by the end of the competition. And so I just want to let you know that this is just the way I've been approaching the problem. It's not the right way to approach the problem, or there's the best way to approach the problem either. If you look at the scores on the leaderboard, there's still a lot of room to go up. There's still a lot of things that we are not doing well at, and we as in the field. And so definitely take all of this with a grain of salt. It's just kind of the way I've been thinking about it so far. Okay, let's get started. So I'm gonna start from the very beginning of when I opened up my phone, heard about the Virtual Cell Challenge, and thought to myself, okay, how am I gonna tackle this problem? And probably I'm thinking similarly to many of you here in this room. The way I was thinking about tackling this problem is, okay, let's think about the model architecture. When dove deep into all the literature, the state model, the SCGPTs of the world, and thought, okay, these are using some fancy techniques. Let me do a little bit better than that. And during my first week, I was iterating over and over again with these bigger models and my own version of these bigger models. Just gonna scroll through. And unfortunately, I made very little progress. It was very challenging. And I was reflecting on, okay, why is none of this working? And I think the piece that I started to understand is that it's maybe worth rethinking about how we approach modeling with this particular problem and AI and bio overall. And that is, instead of focusing so much on the model architecture, I think it's important to think a little bit more holistically. And what I mean by holistically, it means not just thinking about the model architecture, but also the data and the applications and the metrics. And so I described that first strategy as gaining intuition about the data and the metrics. Now, this is counter to a lot of what people are saying in the natural language space. People say, oh, just throw in a lot of your data, everything will work out towards the end. But I think there is a big difference in biology as opposed to natural language, and that's that we don't have this intuition. Like, I don't think anyone has an intuition about what is the right vocabulary for single cell data, or what's the right grammar for single cell data. I'll give you an example of a model that actually does this really well. I think AlphaFold your pinnacle structure prediction or your canonical one for AI and bio. If you look at the kind of the underlying understanding of how they model things, they took a lot of protein sequences, aligned them, and used the covariance patterns to figure out the structure. And so one would argue that they had developed a nice intuition about the vocabulary of protein sequences. There's only a finite number of residues, and protein sequences have directionality to it, so it makes it a little bit easier. They also had an intuition about the grammar. They understood that certain amino acid residues, when they covary with each other, may inform about the structure. And they were also very specific about the application of their model, and that was to do structure prediction. And so that was the first kind of thing to focus on my end, which was, let's try to gain intuition about the single cell data. And I don't really have a good answer of how to do that, but I'll give you an example of one piece that is likely important for gaining intuition about the single cell data. And that one piece is data normalization. So data normalization is required because when we measure the single cell gene expression profiles, there's a lot of other effects that can skew it or bias it. And we want to be able to correct for those artifacts. And so this paper is one that was very instructive for me in terms of, okay, what are the various technical artifacts that we want to capture and that can bias our single cell results? And how do we normalize that? There's one other figure that's particularly important, which is this one on the bottom right, which is depending on how you normalize the data, you get very different statistics. And this highlighted to me a little bit more about the importance of data normalization, the importance of, again, gaining intuition about the single cell data. And so throughout this, I'm going to give you kind of overarching themes, but also specific tips. So specific tip for this strategy one, again, gaining intuition about the data and the metrics. Dig through Arc Institute's cell eval package and cell load package. Those are the tools that they've been using to, one, train their state model, but also evaluate their models. And ask the question, how's the data processed? How exactly are the metrics calculated? The way I like to dig through some of these packages is just using my favorite AI tool. It just allows you to dig through all that layers of abstraction and give you the necessary information really quickly. Also, as an exercise, I'm suggesting to look at differential expression as an example. And that is that connection between the data and the metrics is particularly important. And so when we look at the differential expression, it's taking the data and then analyzing it according to some metric. And the virtual cell challenge uses the Wilcoxon test. So one thing to think about is, how does the Wilcoxon test capture biology, and how does it capture technical effects? So onto the second strategy. One thing that I also struggled with a lot during that first week as I was working on these bigger models is just that I would run these models, then I would actually physically go outside and go on an actual run, and then come back and it's still running. And of course that's a tendency for these bigger models, and that's important, but I also felt like I was not gaining intuition fast enough. And so the second strategy is to maximize human learning in addition to the machine learning. And the basic strategy there is make sure the models are running quickly and efficiently. So one example of how the speed kind of matters is as I was iterating with faster models more quickly, I was able to do a lot of comparisons between model 1 and model 2, dig into the actual data that it's predicting, dig into how each of the models are performing across all the perturbations, and ask myself, well, why is model 1 doing better on gene X for perturbation Y, and model 2 doing worse for gene X on perturbation Y? And start to develop some positive controls of signals of, oh, this is something that both models should be good at. Why isn't that being captured? And so again, the trick there isn't so much the strategy or the models themselves, but it's actually the speed of iteration. And again, the goal is to maximize human learning in addition to the machine learning. And so specific tips for this strategy. explore the variety of tools that are out there to optimize speed. And so I've listed a few on the right here for specifically single cell data. SC dataset, BioNeMo, which was developed through NVIDIA, has specific single cell packages as well. And then Splode is one that I've written. It's a very tiny snippet. I put it on the Discord channel for Virtual Cell Challenge, so feel free to try using that too. I don't think there's a single one that's like the one to use. I think they each have different purposes, and it's worth experimenting and see what works for you. The other specific tip is to think about the metrics and whether a faster differentiable version may be helpful. So if you look at across the Virtual Cell Challenge, there are 3 metrics. There's the DES, there's the PDS, and there's the NAE. Some of these are really simple and very quick to calculate, like the NAE. Some take a long time to calculate, like the differential expression metric, DES. And so think about, like, is there other ways to speed that calculation up faster, or is there a way to get an analog that's differentiable so you could just sync that with your model itself and use that as a loss function? All right, and the final big picture strategy. During this first week, thinking so much about the model, thinking so much about, okay, let's talk about attention and where those heads are, and thinking deeply about the architecture, I was starting to develop this very myopic view of this problem. And one thing that I started to realize is maybe I should actually step back and think more holistically. And think about, again, not just the data, model, and applications, but all in the context of biology. And that leads me to my third strategy or third thought. And that is that the messiness of biology creates challenges, but the complexity also creates opportunities. And so what does that mean? I'll give you a couple examples to highlight that. The first example is technical complexity. And so the ARC Institute put out this really nice blog post about how they gathered the data behind the Virtual Cell Challenge. Here they used a single cell platform called 10X, and specifically the Flex version of the 10X. And one thing that people who work with biology data a lot understand is that measurement results can vary a lot by day to day. It can vary a lot by batch to batch, experiment to experiment, but also by technology and technology. And so that's part of the complexity that's worth keeping in mind. The other example, on the flip side of technical complexity, is the biological complexity. And that is thinking deeply about, okay, what's actually going inside the cell? What is actually producing these outputs that we call gene expression? And when we look at the cell, there's a lot of regulation going on, and that can be extrinsic, so outside the cell, intrinsic within the cell, and even within the cell, there's epigenetic factors, there's what's called transregulatory transregulatory elements, or cisregulatory pieces as well. And so these refer to the mechanism of how the gene expression is actually being regulated. And that is a very complicated process, but it's also worth keeping in mind. And so a few specific tips on strategy 3. Measuring biology is a very messy process. There's no perfect measurement and no perfect analysis method. So there's, we could think a lot about things very theoretically and try to optimize for how we are analyzing the data, but sometimes it's best to just approach it empirically, by observation and by experiments. And here, when I mean experiments, I mean more data analysis experiments or modeling experiments. The second specific tip is to think about this range of biology and technical artifacts. Which pieces of it do you think is worth capturing? I think being able to kind of have that sense of which ones are important, which ones are not, is particularly important. And can you turn that complexity into an opportunity? Can you, sure, when we actually measure we don't really want to measure artifacts, but the reality is that's what we're receiving. Can we predict some of these artifacts, or can we predict some of this complexity and leverage that for increasing our score? And so with that, I want to kind of just summarize again the 3 strategies or 3 high-level messages. One is gain intuition about the data and the metrics. Don't just think about the model architecture, think about all three pieces. Think about maximizing human learning. Make sure you're gaining that intuition quickly, in addition to the machine learning piece. And finally, embrace that messiness and the complexity of biology. It can be frustrating at times, but at the same time, it's incredibly beautiful. And that's what makes this problem really exciting and fun. And this is just the tip of the iceberg. There's so much more complexity in biology to cover. And so I hope you get excited about this and want to explore this more deeply. And definitely keep an eye on who raised their hands on the biology side and make friends with them. Because I think their insights will also be helpful in addition to your the tech expertise that you all have. Thank you.
What questions have for, well,
On your first strategy for the two data sets, cell eval and cell load, you were just cleaning those data sets, but what were you trying to, first of all, what AI tool did you use, and what were you trying to glean from cleaning that data set?
Yeah, so those are actually not data sets, those are packages that the Arc Institute distributes for cell load is to load their data into the state model, and then cell eval is to evaluate the performance of your model relative to another model or relative to a baseline or just on its own. And I'll touch on the cell eval piece. So the thing, I used a mixture of tools, and probably all the ones that everyone else is using. So Claude and GPTs are the ones I'm using quite frequently. But the questions that I'm asking it is, how exactly are they calculating differential expression? What are the normalization patterns that they use to process the data before calculating differential expression? When are they using that? When are they not using that? How do I make sure that my data is going through the right path? And so, the differential expression is just one piece of it. There's also the PDS metric. The MAE one is a little bit easier to grasp.
Thanks for the talk. Can you talk a little bit more about why you think like SCGPT, for example, doesn't perform so well? Because you mentioned in the beginning you tried it. You have intuition for what the failure reasons are.
I don't think I understand enough. To me, yeah, I could give you a lot of guesses, but I don't think they would be all they'll eventually be all wrong. But on my end, it was actually that speed that was the biggest blocker. Like, I feel like I wasn't getting enough of the intuition about the data and the metrics, and I wasn't being able to iterate fast enough across all my models with some of these bigger things. And so I'm not sure whether these bigger models are insufficient, but rather are insufficient for the challenge, but they were kind of insufficient for my first week of working on this project.
Are you building in a validation layer? I know you mentioned one of the strategies is empirical. So how are you able to validate? Because that is the harder problem in biology. Like, how do you know?
Yeah, absolutely. And I think there's one strategy that you can do is frequently submit to the virtual cell challenge leaderboard, but you'll only get feedback once a day, and you have to wait 40 minutes before you get that feedback too, or 45 minutes sometimes. So that's probably not the most efficient way. So I do the usual, splitting up my training set into training and validation or training validation.
Thanks for the talk. So it seems like you do a lot of data explorations. I'm just wondering, do you explore any external data set, or you use purely from what Arc Institute has provided?
Yeah, definitely looked across the world, the landscape of data. There's a lot out there.
Okay.
And a lot of people ask me, is it worth it? And I think my answer right now is, I'm still trying to see. Yeah, it wasn't like slam dunk obvious.
I know you mentioned the three categories that the ranking is kind of evaluate. But do you have more insights on how the rankings are determined, other than just submitting multiple times?
Yeah, I guess maybe my answer there is I would dig deeply into that cell eval tool,
Okay.
because their cell eval tool is very. I believe that's the exact tool they use for ranking at the end too, or giving you the score. So that gives you exactly the algorithm and would be helpful for you.
Yeah, I'm curious about how you see this challenge in the context of developing high quality cell models in general. I guess if this is the challenge that you would hope it would be, and if you were to design a virtual cell challenge, how would you design it?
Yeah, I've been thinking about this a lot. I think, and I was thinking about kind of like the first year of this virtual cell challenge, there'll be subsequent years. Like, what are we going to take away from it at the end? Like, are we going to take away from, at the end of this challenge, is someone going to release like an AlphaFold that's, oh, this is the gold standard and everyone's going to use it? I actually don't think that's going to happen at this first year of this challenge. I'd love to be proven wrong, but my guess is what we're going to all take away from this first round of virtual cell challenge is, what are the right metrics to use? What are the right metrics to optimize? Where are we hitting on the scaling laws of data? Like, how important is generating more data? Is it important to generate more data on the scale of more cells? Or is it more important to generate data across the scale of context? Or maybe across the scale of technology? I think those are going to be really important questions that will be uncovered during this virtual cell, first year of the virtual cells. And I guess the third piece is getting everyone excited about this, getting excited about this problem. And I think that's already been a success, so we're already knocking out that one piece. But I believe the conversation will also arise on the first two pieces.
Maybe a follow-up question, I guess. What data do you currently see would be sufficient to give you a chance of creating this quote-unquote AlphaFold cell simulation, which I presume is you can publish like a tremendous amount of useful generations from the model is being trained in this data set?
Yeah, well, given the scores that are on the leaderboard right now, I think all these models are struggling, right? Even at this particular task. And so my guess is that, for if you're a pharma company or a company that's trying to create a model to solve your specific problem, the best bet still is to train, to gather that data that's very tailored to your problem. So in the virtual cell challenge, it's H1 embryonic stem cell line. And what kind of data to gather for that is still a little bit uncertain, but I think it's important to capture the context that you want to apply your model to, as opposed to just trying to get as much data as possible but while being agnostic of the application. Yeah.
Any more questions for you? Okay, one last round of applause for you. And we'll go back in conversation with you sessions.
Fantastic.
Great. So up next, we are graced with the presence of the fourth place submission at present on the challenge from Markov Bio's Adam Green. So not only is Adam in fourth place, but he's the founder of Markov Bio, which has created high-quality interpretability outcomes from a lot of these models. Put out the first high-quality interpretability model for single-cell modeling. Let's hear a warm round of applause for Adam.
Yeah, after we can generate, yeah.
Cool. And you got me on the Zoom?
Hi, I'm Adam. I'm the founder of Markov Bio. We're building interpretable biological simulators. I don't know if we're still fourth. We put up submission to VCC challenge like a month and a half ago. I haven't really checked it much since. We'll be putting more effort to it soon. Yeah, but for context, I'm an ML guy and a bio guy. I've been working on so-called virtual cells for a couple years. And really actually, I think good enough. Really the problem I'm fascinated by is how can we increase the rate of biomedical progress, and what are the rate limiters to that? And it seems like, with the rise of AlphaFold and structured prediction models, we are getting very good at drugging things or designing very small things to bind to particular other things. But the problem of, what to drug or which patients will respond best to it is still largely unsolved. And this is in spite of the massive amounts of data we now have on these sorts of biological problems. So it raises the question, are we on the precipice of some kind of, say, AlphaFold-like moment, but for the question of biology in the larger systems than single proteins, like we saw with AlphaFold, almost maybe 10 years ago, 8 years ago. So a virtual cell might be the thing that unlocks this. A couple years ago, when there's the first wave of excitement about virtual cell models, but by which I mean, foundation models trained on gene expression data, like the sort that you'll be using in the Virtual Cell Challenge hackathon. I trained the model on this sort of data a couple years ago. I found some interesting things, did some kind of proto-interpretability, but not much came of it. And recently, say within the past 6 to 9 months, virtual cells have come into vogue. So it's quite an exciting time. But there's a question, what do we mean by this? Is a virtual cell just a simulator of experiments we'd otherwise run in the lab? Is it a specimen, something to be studied and investigated like we might a real cell? Or is it some secret third thing? And so I think where my definition of a virtual cell departs from how most people in the field think about it, I think yes, you want to be able to simulate experiments. Say, given a cell type, given a perturbation, whether that be a gene knockdown, a small molecule, say what would happen to the cell through time, maybe in the length of gene expression, but eventually in other kinds of biological modalities. But I think even if you had this sort of model, like a perturbation prediction model, it still wouldn't be all that useful for doing things in biology, like real tasks we care about. So rather than just treating it as a black box, what if we had the ability to peer inside it, see what was going on, maybe develop interesting hypotheses about how to control the system. And so this is the realm of interpretability, and so I'm going to talk a bit about to give you inspiration for projects you might do today. And for context, we've gone very deep in training these models. It's a difficult task. I wish you all well with training them today, but I'd encourage you, if you have an interest in interpretability, this may be another path to explore, and you can create some differentiated, interesting work that other people are not working on. It might be a bit more feasible to accomplish within a day. So around 9 months ago, we trained one of these virtual cell models from scratch. We did some of this interpretability work on it using what are called sparse autoencoders. So if you're familiar with Anthropic's Golden Gate Claude, basically you have this model, you run a bunch of forward passes, you collect activations, then you train a sparse dictionary, which is like a list of features or concepts inside the mind of the model that it uses to understand the biology it's simulating. And then we went about trying to label these and see if they recovered known biological concepts. Here's the basic math framework for training an SAE. And so when we did this, we recovered a bunch of pretty interesting concepts and put some of them out in this kind of feature explorer. So to give an example, within this sparse feature dictionary of our SAE, you look at a given feature vector, and then you say, which cells does it tend to activate on, which genes? If I turn it up, what happens? If I turn it down, what happens? Which other features downstream does it seem to regulate? And so we found a variety from features that fire in, plasma cells, to some kind of drug metabolism or general hepatocyte metabolism program. And then you can do interesting things like cluster these features and say, do they tend to fire together? Are there high order concepts inside the cell? And then you can do some kind of, PageRank type algorithm on these internal feature circuits to identify what might be important features in any given cell state. So for instance, looking at this set of thymus cells, and as they differentiate into different immune cell fates, we identified certain features that appeared to indicate, what cell type it was, or if it was going to go along a particular trajectory or not. And so, for instance, we recovered, using this kind of PageRank type approach, this known transcription factor that upregulates on a certain T cell trajectory, which is interesting. But moving beyond single cell, we then look, could we identify interesting spatial patterns and how these features fire? So in this case, we're looking at, I think, a 2D slice of intestinal tissue. And can we pick out, unique populations of, say, enterocytes or goblet cells, these cell types in the intestine? And so we found a feature that was highly enriched among the enterocytes that also appeared to activate pretty highly among neurons. It might be some kind of conserved program related to vesicles, moving between cells. So all this to say, when you train these sorts of gene expression models, yes, one application is, okay, can I predict the unseen experiment? Other interesting application, and probably what will be necessary for these models to actually become useful, is saying, how does this model make that prediction? And perhaps if, by learning to make the loss go down, it actually recovers real laws of biology, or a true world model of biology in the cell, then by probing that, you could do many interesting things. And so here are some possible applications. I don't think you'll accomplish these within the hackathon today. Drug discovery is a long process, but maybe you come up with an interesting hypothesis. Target ID, I have a disease, which target should I hit? Or I have an existing target, if I hit it, will it work or not? Mechanism of action elucidation, so I have a drug, it has an effect, but I don't know how. I want to figure out how. Or patient stratification. So some patients respond, others don't. You have a tumor, it's resistant to one drug, another tumor is not, and it's susceptible. Why is that? So for today, what I'd encourage you to do if you wanted to explore interpretability projects, take an off-the-shelf gene expression model, like SCGPT, or state model, and then try exploring some sparse autoencoder frameworks. So NInsight is a general really good library for doing activation caching and other things with models, applying internal perturbations, if I want to ablate a feature, if I want to upregulate a feature or an activation. Feel free to explore that, it's extremely flexible. And then Eleuther is a sparse autoencoder library which allows you to, say, take activations that you grab from a model, train on one of these SAEs, one of these feature dictionaries on it, and then you can do really interesting things. So for instance, when we trained our SAEs, the previous ones 9 months ago, you get these features, you say what cells do they fire on, what genes, and then we fed it all into Claude and said, can you propose a label for this feature? So I'm not going to go through 16,000 features and hand label them. And Claude has a surprising depth of biological knowledge. So if you do train this kind of SAE, maybe you can say, hey Claude, what do you think of these features? And then to zoom out and kind of motivate why this is an interesting problem, and Jeremy talked about this, it seems like we're on the precipice of some kind of GPT-1 or AlphaFold moment of biological simulators. And really, virtual cells are a small step. Now that we have this abundance of data, you can ask, sure, modeling a single cell is cool, but what about modeling multiple cells? If cells in some kind of 3D configuration, or eventually even the organ level. And given this abundance of data, just to show the richness of data that we're now able to collect, this is, I think it's light sheet microscopy data from CZI on a developing zebrafish embryo, where each of those little dots you're seeing is a little cell. Hopefully this pumps your intuitions on where these models could go and why they could be important. Biology is going to be an extremely data-rich domain. It already is a data-rich domain, and so given that, seeing what has happened in the world of LLMs, you might say, maybe this will be amenable to the scaling approach. Markov were of the belief that people are not taking scaling seriously enough, certainly in the field of virtual cells. And over the next year or so, there's going to be 1 billion plus cells coming online. People are going to train interesting generative models of these data, and there's a question of what they'll be able to do. So we're pretty scaling build at Markov. We're training the world's largest virtual cell model. We're gearing up for a 10 of the 25 flop run within a year. We're hiring. Come and ask me about biology. I have lots of thoughts on how to train these models, so I can provide some guidance. Yeah.
Questions, Fred?
So are you focusing the simulation approach more from developing a model itself around, say, a particular cell or cell structure, morphology or the expression, or are you looking at much more generatively? when you're thinking about virtual?
Yeah, more the latter. And just some points of clarification, people, in the current context when we're talking about virtual cells, most people are referring to, models of single cell gene expression data like this, what you're dealing with VCC. In our case, yeah, we take a more of a generative model approach where you're just basically training a big LLM to predict the masked or some other loss gene expression token. But there's certainly other approaches you can try. I mean, like Rita talked about, maybe that's not the only approach, and maybe not the approach you want to take today.
Just sort of a biology curiosity question. In your modeling, have you found the predictions more accurate for the next, 10 seconds versus the next 10 hours?
Yeah, so this actually gets at one of the limitations of these data. So in the VCC challenge, you'll have, a bunch of rows, which represent cells, a bunch of genes, which are the gene expression values, and each is associated with some metadata. Is this a control cell? Is this a perturbed cell? What target gene did you perturb? The issue is we currently don't have, or there aren't really great high throughput methods to longitudinally profile a cell over time in RNA space. There's some ways to get around that, people are trying, but really you're dealing with destructive measurements, snapshots in time. It would be great if we had that sort of data, and I think it's probably going to take some combination of, high feature density RNA-seq, maybe some kind of non-destructive imaging or another measurement technology to get that sort of model one day.
I'm curious about like 3D dynamics and 3D modeling, and where you see like the most value being. Obviously, even in the context of image generation and video generation, 3D has been a challenge in ML. And yeah, you have to hear is 2028. So I guess
Maybe slightly ambitious. We'll see.
Yeah. So I'm just curious to hear, do you see any interest in 3D data at present, and do you see that as being like the target of data creation efforts, or is that kind of premature?
Yeah, I think the timing is actually quite good. So really the inputs to getting these data are, you need the actual biological systems. Can you culture the organoid, like this 3D amalgamation of a bunch of cells? There are tons of companies spinning up data sets for this. But really the expensive part is, how do I measure the signal from that? And the inputs to that are sequencing costs and these library prep costs. I think the data is going to be there within a couple of years. There are already vendors from which you can buy 2D spatial omics data. It's like relatively cheap, and people are starting to collect more of it. To then go up to 3D, it really is just a matter of putting resources into it. And I think the timing should be quite good. If we build a good virtual cell model of an individual cell, hopefully that can be used to initialize like a good multicellular model, in the same way that having a good image generation model would be a useful initialization for a video model. That would be the hope.
Can you say a bit more about your performance on the virtual cell challenge, and what was involved in getting the score that you got?
Yeah, so we threw up an attempt like, I don't know, a month and a half ago. Like I said, we're probably going to put more effort in over the coming month and see how we can do. We took a pretty scaling build approach where we pre-trained a general model of the cell on a bunch of other gene expression data, and then we fine-tuned it on the 150 perturbations in the train set, and then evaluated it on our ARCS50 held out validation perturbations. Really it was just a bunch of ML engineering and research, and then some kind of domain-specific insights on how to train these models, where we differ a lot from other people. But fundamentally, like at its core, it's just a kind of LLM for gene expression tokens.
I see. So you feel like the pre-training did a lot of the lift in terms of the performance?
Oh yeah, I think it did all of the lift. We can compare. If I take the same model, randomly initialize the weights, train it from scratch, versus I take the pre-trained model and then fine-tune it, you see a pretty big delta in performance. And we hope that'll increase as we continue to scale the models on the pre-training side.
More a general question. So I worked in one of those CROs for 10 years. We've seen drugs pass and fail. So so there are two drugs that pass. So in 2008, you have Tysabri from Biogen, which is a monoclonal antibody. Okay, incredible results. Like before that, we only had interferons, and now we have this monoclonal antibody that works. And suddenly, after a few years, people started dying of JC virus. You know? They were like, oh shit, again. Then they removed it from the industry. 10 years later, somebody figured out, oh wait, I can have a monoclonal antibody, but it needs to be more specific. I need to target B cells, right? But that intuition only came because those researchers are looking only at the immune system and only at one disease. How does all this, like the virtual cell fit in? Like, do you focus on one disease, Alzheimer's, MS, just want to hear your thoughts on that.
Yeah, super interesting. I should say, patients, like whole human beings, are extremely complicated. There's a lot going on. In the history of drug discovery and development is riddled with these cases of where we have a simplified model of human biology that ends up getting overturned because we find out it's a bit more complex than that. Yeah, in the case of B cell specificity, like how would, an interesting question to ask is, had we had a virtual cell at that time, how could it have avoided these sorts of issues? I don't know. I think the models might be a bit premature for these sorts of questions. Certainly, I think you can say if I know that this is the correct, specific target B cell I'm trying to deplete or affect, like maybe I have a way of stratifying patients based on some kind of virtual cell embedding information from their, blood I collect. Really, the claim is that for these to become truly therapeutically useful, you're going to have to make this move, from a single cell to multicellular to modeling bigger and bigger context, so you can start to, predict some of these events that maybe happen, multiple years out in the patient.
Any more questions?
I have more of a piggyback comment on that. So just a sort of primer for anyone that's not super biology. It's not uncommon that we find cell lines in a dish that die in response to a drug, and then you take that same exact cell line, put it into an in vivo context, and you see nothing happen. So basically all the data that we have here is from that in vitro environment where the baseline molecular features of that cell are identical, and you change the environment that it's in and you get a completely different result. And you see the similar things with efforts like DepMap, where there are effects with what media you were using for your cells. Completely changes what will and won't kill you. So just a little bit of temperament on how it's more a temperament on the unseen and unmeasured variables that are in this very complex nonlinear equation.
That sort of is part of my earlier question, is that we're basically being evaluated on how we predict that particular cell line with that particular 10x machinery.
Yeah.
So it may not be, that's why using other databases may not help the model. Is that?
Those other databases are also from a completely different technology.
Exactly. So it actually might make your model worse if you pre-trained on other data, possibly.
In my view, it depends on like the coverage and overlap that you have. Like if you have technical replicates that that came from the same exact single cell suspension, and then you did the there's probably a learning function that could be learned to translate, but the diversity of space, like cell state space, that you'd need to cover for to build a comprehensive map across those topologies is going to be a very large space. So you'd need probably a similar effort of this scale, in my view, to be able to have a good control for that. And it would have to be really well controlled where it's actually technical replicates, like triterate your cells up and down, then that goes to that technology, that goes to that technology. But yeah, no, it's very good points and challenging.
Yeah, just to provide some commentary on that, I agree, being able to model a cancer cell line in the dish isn't going to get you to solving cancer in a real patient in vivo context. But the hope would be maybe to provide a counter to this more tempered view is that protein structure models are modeling static structures. And getting that sign of life with AlphaFold 1, that this approach could scale up. led eventually, you know, to all these, the blooming of all these other efforts of structure modeling, that I think eventually will go, you know, capture the real messy, dynamic protein processes that actually unfold in the body. And maybe there are cases where you can get a bunch of utility even with really primitive models early on, but I agree 100% that like data quality is fundamentally the rate limiter here. If you want to simulate a human being, you got to collect measurements of a human being in different states. We currently don't have the technology to do that in a really high throughput, cheap manner. But you know, being able to measure a single cell is a good first step.
Cool. Thank you. Fantastic. Thank you to Adam with Marco Bunny.
People pitched that to me as my postdoc. Incredible. I said,
Wonderful. So our next speaker is maximally counterfactually responsible for the existence of this event. Many thanks to Yusuf Rouhani for being with us. This is the first author behind the paper, behind the Virtual Cell Challenge, and his team has done SC base count, biodiscovery agent, created state, the model that they sort of shown how to train on the Virtual Cell Challenge, and of course created this challenge. So yeah, we look forward to hearing about it. The ARC Institute, which was created by Patrick Su, has also been doing phenomenal work in genome generation. I think it's a real phenomenal moment to be at the intersection of AI and bio, in part because the ARC Institute is doing all of this phenomenal work. So it's a joy to have him. Let's hear a warm round of applause for Yusuf.
Thank you. Thanks. Great. Hi everybody. Thanks for coming, and apologies for being a bit late. It's tough to. I have 2 kids, so Sunday's a bit rough. Great. So my name is Yusuf Rouhani. I lead a machine learning group at the ARC Institute. Thank you, Jeremy and Akira, for this invitation. I don't have a very formal set of slides. I'll just kind of, I guess, broadly talk about what we've been doing at ARC, how we thought about this competition, and I will probably pause at that point and see if we want to go into more other projects, but I guess most of this I'll just focus on the competition and what we're thinking about the virtual cell. And also happy to do this as a discussion. I can stick around for an hour or so later if folks want to chat informally. Great, so I always like to start with AlphaFold. So I guess you can't see the title. Should I share again?
Try Control minus and just shrink your screen.
Does it expect certain dimensions? It's still later to, it still tries up to the, Sure. Yeah, well, yeah, perfect, thank you. I'll try to be quick. All right, so, okay, well, I'll try to be efficient. So everyone's familiar with AlphaFold. What are we doing with AlphaFold? You take a protein sequence, you learn a machine learning model that predicts for you the structure of that sequence, And why this is powerful is because you could have trained on a very small subset of sequences, but you can make predictions for a very larger space of sequences. And in a sense, since each of these, the inference of each of these structures takes various different experiments in the lab, costs a lot of money and time, in a way AlphaFold is simulating that experiment for you. And by doing so, it's allowing you to kind of search over this broader landscape of possible experimental outcomes. So one way to think about this is it's in silico, running an experiment for you in silico, computationally. And this kind of traversal over landscapes of possible outcomes is a very common template that we see in scientific discovery, where you essentially are kind of doing a search, right? Where you have various different hypotheses. You need to find the right ones to experiment and test in a lab. But it's expensive to test out all of them. And so what you end up doing then is you can use computational tools to help you accelerate that search and find interesting regions in that space to dive into further. So what we wanted to do was kind of take this, the power that AlphaFold and ESM and other models give you at this molecular scale and kind of abstract it to the level of cells, right? So so far we've been talking in terms of DNA, protein, and so forth. How do we bring that up to think of the entire system of the cell? That's of much more interest to, of course, therapeutic discovery and various other biological applications. So can we move one level of abstraction up? And that's really been the main motivation behind the virtual cell model is, can we create an AI model of cell state? Can the same way that we've been able to create these models of proteins and now I guess also of DNA, can we begin to model how cells are changing under different conditions, under different time points, and then use that to better inform our experiments in the lab? So an example of that would be, say you have a model of cell state that can capture different states of neurons. So you have a functional neuronal state, you have a disease state. Could you then identify interventions or transitions within this space that could take you in a direction away from more disease to more functional states? And then of course, what's the vision here? If you see this kind of search and experiment paradigm, so really what we're kind of moving towards is more this kind of closed loop framework where you do have a working virtual cell, it provides you with hypotheses, you perform those experiments, and then those experiments and the data that you generate further enhances the understanding of the virtual cell. And so for us at ARC, really, we've always thought of this virtual cell project as not just the models themselves, but also how we generate the data, and how that data fits back into the model to improve its performance. So it's really kind of that entire life cycle of not just model development, but also generating the optimal data to get us to where we need to go. And so just a quick snapshot on how my team has been looking at the virtual cell at ARC. We've been kind of dividing up our efforts into these two kind of broad buckets. On the far left, on the left, what you see is essentially our efforts towards learning a cell state model using artificial intelligence approaches. And on the right, what you see is how we can also use AI, but on the data generation side. So thinking about how can we better curate existing data, how can we leverage agents for helping us design experiments and generate more perturbational data sets. And so really kind of thinking about this whole framework from start to finish. Today I'll be talking a bit more about this side, which is thinking about how we evaluate these models, and this is where the virtual cell challenge fits in. But if you're interested, we've had papers out in each of these areas this past year, and we have more coming in the next couple of months. So happy to chat and discuss if you have questions about any of these things. So just a quick highlight on each one. I think about 2 months ago, we released a new perturbation prediction model called STATE, which uses a transformer over sets of cells. So that's some of the first work we've done on the model development side. On the model evaluation side, we've been working on the virtual cell challenge, which of course I'm sure most of you are already familiar with. And then using AI agents, we created what I believe is the largest agent created biological repository as of right now. And it is about 5 times as large as the alternate existing repository of single cell data. So it's about 500 million cells at this time, and it's still growing. And then finally, we also used agents for trying to design actual perturbation experiments or CRISPR screens in the lab, and we showed that this actually improves our efficiency in being able to detect interesting effects. So that's another paper that we're also building on at this moment. Okay, I will pause very quickly if there are any high-level questions on this so far. Yes.
When you're saying that you're doing a model development,
Yes.
what does the model entail at this point? Like,
Yes.
what are you really collecting?
Yeah, I'm gonna go into that
Okay.
very soon. Okay, so I think we should just keep moving then. So let's actually jump this slide. So what does the model do? So before talking about like what our model does, I always like to preface it by saying that we are not the first people talking about virtual cells. This idea has existed for a long time, for decades actually. Some of the first work in this space was done like early 2000s by professors at Stanford. So Marcus Covert had a very cool model that essentially used series of differential equations to model the interactions between all the various different components and organelles in the cell. There's work from Uri Alon's group at the Weizmann that looked at differential equations for modeling, the dynamics of various different biochemical processes. So this is really a vision that we didn't kind of propose. It's actually been there for a long time. And for very similar reasons, can we use computation to essentially tell us how cells would respond to interventions we've not actually done physically, but potentially we can use these underlying physical processes and laws to guide us in predicting what the outcome would be. And what's changed, and this is something we outlined in a perspective we wrote recently, is that we now have this kind of foundation model approach to looking at biology in various different domains, driven by, of course, the growth in AI and associated kind of machine learning architectures. What folks have now found is that you don't have to essentially create a model for every specific system. You can actually create a foundation model trained more generally on large amounts of data and apply that to various different downstream tasks, many of which you may not have directly trained on. And so the analog of this in biology is, say, you train on a lot of data from single-cell RNA sequencing, from cells that may or may not have been perturbed from various different contexts. And by doing so, regulatory and other kind of expression-based information that you can then apply to different downstream tasks, such as predicting perturbation effect or guiding experiments. So for us at ARC, and I think this comes to your question, we focused on kind of drilling down on one specific task in particular, which was, can we build a computational model that can predict cellular response to perturbation? So again, there are various different approaches to look at virtual cells and what tasks they can do or what data sets you can use. We were very focused on this specific task because to us this captures quite nicely what most people envision when they think of a virtual cell, which is, oh, I'd like to be able to poke it and prod it and see like how it responds to that, and really kind of think of the cell as this this machine that you put different inputs into and you see kind of what the outputs are in response to that. So quite simply, you can write this as a simple equation that takes as input the unperturbed state of cells, the applied perturbation, and predicts for you what the post-perturbed state of that cell would be. So for this specific equation over here, we can also break it down a bit more deeply. So how are we thinking about basal cell state? As I mentioned, there are various different ways of measuring cell state. You could look at transcriptomics, cell viability, proteomics, cell morphology. For us, we were very focused on transcriptomics, given the amount of data that is now available. I mentioned to you, we have 500 million cells now in base count, and combining this with Tahoe and other data sets, we're reaching in the 600 plus million cell range. So it's a really exciting time. As I think some of the previous talks also mentioned, there's a lot of potential here for scaling. And also at ARC, we are very excited about generating our own data sets, and we think that's going to have an even more dramatic impact if we're able to control for all the various different confounders that are present in public data sets and really kind of allow the model to focus on more stronger biological signal. For those of you who haven't worked with single cell data, essentially what it looks like is this. So single cell gene expression data, you have like various different cells here on the y-axis, and you have different genes expressed here on the x-axis. Essentially what you've done is you've counted the number of transcripts that you're seeing for each gene within a specific cell, and you've created this kind of distribution over various different genes in that cell. And then as you compare this distribution over expression in each cell, you can get an understanding of what state that cell might be in and how that differs from other cells. And also you're interested in, if I were to perturb or add a drug, how would this kind of collection of numbers change? How would this expression state change? That's kind of in a very bare bones level what we're doing with these gene expression prediction models. And that's very different, as you can imagine, from say something like protein sequences, where you don't have this kind of like stochastic measurement of expression. You really have a very fixed kind of sequence-based representation of proteins. And so there's a lot of challenges in that sense around how, you know, when people say, oh, this worked for proteins, why does it not work for single cells? I think there's a lot of challenges around the machine learning side. How do we really capture this more noisy and stochastic state of cells in a foundation model in a way that allows us for scaling, as opposed to say more, a more well-defined protein sequences, for instance, that where the signal is much stronger in many ways. So I think these are some subtle challenges that will emerge as you start to work on this problem, and also that we've seen when we were building our models, especially state, which focused a lot on some of these aspects. But yeah, happy to chat more about the mess that single cell data can be. All right, one last thing before I can stop for more questions. So how are we perturbing cell state? So here we can, of course, also look at various different forms of perturbation. For us at ARC, we focused a lot on genetic perturbations, so CRISPR, gene editing based, but you can also do drug-based perturbations. Again, our motivation for that is there's a lot of great Perturb-seq data available, which we wanted to use for training. One last thing, how are we thinking about the machine learning task? So there are actually many different axes along which you can make a perturbation prediction. So one way to think about this is that say you have various different perturbations that you've performed on a cell and you've measured data for that, then you'd like to predict what would happen in the case of a new perturbation. And this is what we call the perturbation generalization setting. Another way to think about these problems is, say you have certain contexts that you've seen perturb at the time of training. So you may have seen, stem cells, you may have seen macrophages perturb at the time of training, and you're interested in how, the H1 stem cell line would behave upon perturbation. And so here you're really kind of thinking about this context generalization task where you've seen these perturbations applied before, and you're interested in being able to predict how they would do in a different context. Why would this be interesting? I mean, if you do this well enough, you could potentially be able to generalize across donors. If, say, you're performing some T-cell therapy and you have data from certain donors, you can potentially predict responses for a different donor that you don't have actual perturbation data for. Okay, so I'll pause here a little bit. I think this is a source of confusion going into the competition, because I think some folks looked at the problem from a perturbation generalization setting, which is totally fine, even though we framed it in the context generalization framework, just because we thought that most models would probably be more effective in predicting responses for perturbations they have seen before. So that's how we kind of tried to frame the problem for the virtual cell challenge. And so you'll see a lot of that terminology when you look at the web page for the virtual cell challenge. But it's totally fine if you actually focus, if you just treat it as purely looking at the training set that you're provided and training just off of that. I think there's already quite a few submissions that have tried to do that and have done reasonably well. Our expectation with kind of setting it up in this kind of context generalization setting was that folks will try to train their own model in-house and then apply it to the data set that we've provided through the virtual cell challenge. And it sounds like in some of the previous talks that is what some folks have done. So it's kind of preempting that question, and so I just thought I'll mention that as well. Okay, I have like a couple more slides. Do I have a reasonable?
Yeah, people are good.
Okay. Let me also just talk about the metrics since we're on this subject. So for the virtual cell challenge, you've probably seen, maybe if you saw the commentary, we had like 3 core metrics that we focused on. The goal behind these metrics for us was we wanted to basically capture, both be like maximally expressive, like something like a mean absolute error, but also capture things that actually matter to biologists. So when biologists look at Perturb-seq or gene expression data, they're often most interested in what sets of genes are most differentially expressed, because that they can kind of interpret into specific pathways, specific biology. And so we wanted to create a metric that captures some of that aspect, and so we created this differential expression metric, which is on the far left. And in the middle, we kind of have this one metric that is both meaningful biologically, but also actually quite challenging to perform well on from a just general machine learning perspective, where essentially what you're trying to do is you are comparing the prediction that your model makes relative to all the possible perturbation outcomes that you've seen in your training set. And I'll show you in the next slide how exactly this is computed. But essentially you're trying to see, relative to everything else, are you able to predict that your response is closest to the ground truth response? So you're trying to essentially learn the subtle differences between all the different perturbations, and not just be able to predict, like, say, a general effect that would work for any prediction. You have to learn to be able to discriminate between the various different perturbations. Okay, so I can do a quick deep dive into each one of these. So just diving a bit deeper into the perturbation discrimination score. So the way to think about it is essentially, say you have your prediction shown here as this red dot, y hat underscore t, and you have a ground truth, which is shown here as this green dot, and you have these various different ground truth values from your training set, which are shown as these blue dots. What you want is that your green dot is as close as possible to the red dot, and most importantly, it's closer than any of these blue dots. So meaning that you want the ground truth to, relatively speaking, be closer to your prediction than any other perturbation outcome. And so then your model has learned to kind of learn the relative perturbation effect compared to like all the other perturbation states that it could have predicted. And then we essentially assign numbers, we assign ranks to each of these. So in this case, you're closest to this one, so this gets a rank of 0, this gets a rank of 1, 2, 3, 4, and then essentially you normalize the rank of the ground truth by the total value, and you get your final score. I think this is actually, yeah, this is correct, yeah. Yeah, so this gives you your score. And so ideally you want the score to be as small as possible, but for the competition, we've inverted it, so you want it to be as high as possible, but 0.5 is random.
What's the denominator?
The denominator is all the possible perturbations that are in your training set. So if there's 4, you're dividing by 4. Yes?
Is there any weighting given to basically the similarity in the ground truth treatment output vectors? So essentially, if I predict that I am in, a state that's close to the ground truth state, but in terms of the perturbation state that's applied, that's a very crowded area.
Yeah.
Like, how do you weight that versus, someone else's prediction, not mine, because mine would obviously never do this, but
Yeah.
it's all the way on the other side of perturbation space.
Right.
Where if it's a very dense space, you may
Yeah.
have a low rank
Yeah.
for that particular comparison
Yeah.
versus something where if you have a uniform distribution of perturbation space, it'd just be
Yeah, so first of all, there are failure modes for all of these metrics, and I think Rio kind of mentioned that too. Like, this is, we expected, we expect to like iterate and find ways to improve these. However, for that specific case, we did actually look at the distribution over various different distances. So we used different distance metrics. We tried different distance metrics. So when we looked at the cosine distance or the Euclidean distance, and I think this is in the state paper where we introduced this version of the metric. The original metric actually was introduced by Yan Wu et al. In a different paper for TurBench. So what we found was that the L1 distance was fairly well distributed. There wasn't a lot of accumulation in one portion of the distribution. So I think for the most part, we've seen a good spread. But we cannot, you're right, we cannot fully correct for all possible failure modes. And it is possible that you could fail that. However, I mean, it might also be a nice way to differentiate between models that are like really doing well. So there's always a trade-off here. Yeah, that's a good question. Sorry. Should I keep moving?
Yeah.
Okay, yeah, we can come back to more questions. So the differential expression score, what does this mean? For those of you that haven't heard of this term differential expression, one way to think about it is you have cells, actually this is a little confusing. So you have, let's just ignore this. You have cells and you have their gene expression values. You look at the distribution of the expression values for each gene, and you essentially compute how much of a log-fold change are you seeing between the perturbed and unperturbed states. So this could be the predicted perturbed state or the ground truth perturbed state. How much of a log-fold change are you seeing, and what is the significance value of that shift? So maybe I'll just go to the next slide, so kind of it's clear. So here, essentially, so you have each dot here corresponds to a specific gene. And then on the x-axis, what you're seeing is for as you compare between the perturbed and the unperturbed state, what is the log-fold change in their average expression? That's what you see on the x-axis, and the y-axis is capturing like how significant was that change from a statistical perspective. So you have the log 10 p-value, the negative log. So one way to think about this is the farther right you are here, the more you are overexpressed in the perturbed setting, and the farther left you are here, the more you are underexpressed compared to the control expression. And the farther up you are on this plot, the more significant your change was, statistically speaking. So why would that be important? So say you have very few cells, you might measure a very large log-fold change, but in terms of statistics, it might just be very insignificant. So then you don't want to count that too much. So that's kind of the way that people think about differential expression. How did we apply this to our metric? So what we did was we thresholded both the model and the ground truth values at a specific significance threshold. And then we picked a top n number of differentially expressed genes for in ground truth and n number for the predicted model. And essentially we compare the overlap between the DE genes that the model predicts versus the DE genes that you see in the ground truth data. And the more overlap you see, the higher your score is. And that's kind of how we designed the differential expression score. And I can tell you more about, I guess, how the n number is determined and other things. But does that make sense? Any questions on?
So how much of the score is critical into the overarching model that you're actually developing? Whether you're applying that for training data set that you're actually looking for this score and ranking?
We just use it for evals. We don't use it to train on, yeah.
Okay.
Yeah, but I mean, it's a good question. Like a lot of the losses that most models train on don't always correlate with some of these scores. So it's, yeah, it's an open question. How do we, on both better designing the metrics and also better designing loss function. Okay. I think, so I'll just kind of wrap up a little bit now. And so the last score is, of course, the mean absolute error, which I don't think I have to explain to anyone. Essentially, what you're looking at is the raw counts, and you're comparing the raw counts with the ones that you predict versus the ground truth value, and how off is your prediction. So this is the most standard machine learning performance metric. Okay, if you're interested, do check out Cell Eval. We have a bunch of other metrics on there as well. This was part of the original state publication, and you can probably use those as well for diagnosing how your model is doing. Yeah, and yes, and then finally, of course, yeah, there's a lot of activity. I took the screenshot this morning. The leaderboard seems to be changing a lot, so you probably already know this. There's a lot of money involved, a lot of people are participating. So it's, yeah, do try it out and feel free to reach out if you have more questions. I'll skip this part. It talks a bit more about the data generation procedure. Yeah, and then if you're interested in any of these other projects, maybe offline, I'm happy to chat. We're really excited about this direction, and we're working pretty hard on both, as I mentioned, developing models as well as generating the data. We have 2 open positions right now in my team, one at a more junior level, say like straight out of your PhD, and another one that's a bit more senior. So you can reach out. Also, we do take in interns as well. If you don't have a PhD, don't worry. We do have like temporary positions that we also do. So yeah, just email me. Yeah, so I want to thank everybody who's involved in this effort, especially like everyone with an asterisk on their name was a core contributor to some of the state modeling and also developing Cell Eval. If you need links to the papers, you can check. I think they're on my website. And yeah, thanks for listening. Happy to discuss any part of this.
Questions for Isa?
Just curious, how did you guys pick the H1 embryonic cell line?
Oh, that's it's a combination of like inertia. We were already working on it, and
Fair enough.
it's also biologically quite interesting. Like it's definitely more interesting than K562, I would imagine. Yeah, I was pushing for more context for this competition. Hopefully, the next round next year we will have more cell types. Yes.
Why'd you guys pick the flex kit?
Yeah, so we've done, okay, So we've done a bunch of tests, which we wanted to release with the competition, but we were, hopefully it'll come out soon. We compared to 3' and 5'. We found that Flex was more reproducible on its perturbation effects. It's not perfect, but it was doing a bit better. Also, the experimentalists love Flex, because I think it's just like a lot easier to do like larger space, because you can fix the cells and all of that. Yeah, and I think there are a few other reasons as well. I think it's nice to have the probe-based measurements because we found that for some of the more lowly expressed genes, we were doing a bit better with the probes. But yes, you do obviously give up on other kind of intronic reads and things that you don't specifically measure for. But we did make a trade-off, and I think so far we've been pretty happy with it. It's not perfect, but yeah.
Would you guys release the other?
Yes, we will. So yeah, all the data is going to be released. Everything's going to be released. We just have to put together the paper and stuff. So we kind of sort of made progress towards that with this blog post, which provides a bit more of the backstory around how this data was generated. So, but we will after the competition ends provide the data sets. Yes. I already yes. Yeah.
Thank you for the talk. What do you think are sort of the key bottlenecks right now, and like what do you see as the role of integrating other data modalities, which maybe they're like underrepresented compared to transcriptomic data?
Yeah, no, that's a great question. So there's a lot of things that need to be fixed. This is my people don't expect me to say this, but I don't think scaling is enough. Actually, I think there's a lot that actually has to be fixed, and it's really exciting at ARC right now for us because as we're scaling, we're discovering so many things that we can fix along the way. Not just in terms of the models, but also in terms of how we're generating the data, even the assay itself. So we're making a lot of cool changes. Happy to chat more about that. In terms of additional modalities, I think at the moment we're kind of a little more bullish on transcriptomics given the vastness of the data available there and kind of the flexibility and the expertise we have in-house on that modality. So we're focusing a bit more on that. Yeah, I mean, I think at least for the VCC, I don't know if additional modalities will be super helpful, but yeah, I look forward to seeing, I think, models that do more with multimodality. Yes, go ahead.
This might be similar to his question.
Yeah.
If it is, feel free to disregard. But on the perturbation distance calculation,
Yeah.
Why does directionality not matter?
Yeah. I guess. I think we did get this question. Maybe just I think we initially thought that absolute distances would be sufficient. I think directionality becomes a little tricky to capture.
Because then you can see if a submissions model is consistently wrong in the same direction.
Right. Yeah. No, that's a good point. We should try that. We just, I think, intuitively felt that like you might over penalize if you include directionality as well. Yeah, but I don't have a deeper answer on that. Yeah.
How much of the simulated data sets you're also using for this purpose? Are you actually using the existing data sets that are available and validated by biologists and others and utilizing for the purpose of experiments? Or is there a way to just pretty much, since you're using agents anyway in creating data sets, can you actually start simulating and only use simulated data sets to perform?
Sorry, what do you mean by simulated data?
For example, if there's a rare disease, there's obviously not enough data that's available. But if I know the ground truth of just this limited data set, what if I can actually start developing my own simulated data sets, add to the mix and start validating from the existing data set, which is derived from a patient others, versus, that you're using for experiments?
Yeah, I mean, I think that's an interesting research direction. We're not working with any simulated data at the moment, but I think that sounds interesting for sure.
By way of example, for the more old hat AI folks, there was this reinforcement learning kind of Achilles heel that was discovered and went viral by this race boat simulation where the race boat kept going in a circle collecting coins because the respawn for the reward was basically just to maximize your points. So the race boat never actually ran the races, it just kept going in a circle. So as using that as sort of a backdrop, given that we're basically trying to fit the evals, what are your biggest areas of concern in terms of the potential Achilles heels within the eval metrics where we might be trying to fit the wrong thing?
Yeah, no, that's a great point. So I actually don't think if you do perfectly on these metrics, you've like won or like it's solved, the problem is solved. I actually don't think the winner of this challenge would be close to AlphaFold. I think when we first put out this competition, we definitely wanted to start a conversation with the community. When I guess in our paper state, we were like a little more opinionated, like, oh, this is what we think is good. But I think with the VCC, we also wanted to like get a feel from the community, like what's working, what metrics are working, and even what problem formulation is working. Like it might be that this kind of context generalization task is not something that is actually, may say therapeutically or research-wise, like relevant. So I think we're very open to exploring alternate approaches. I think one thing I will say is something we're doing a lot more of at ARC as well is making sure that we're moving past just these machine learning metrics to thinking about what is the actual like how are you pushing the bottom line in the lab? Like, are you improving efficiency? Are you improving the time it takes to get to an interesting hit? That's an interesting example. Another thing we're looking at is also moving evals past just looking at Perturb-seq, because Perturb-seq and single cell data comes with its own bag of issues. How can we kind of create evals that don't rely on just single cell data? So there's a lot of directions we're thinking about it. Yeah, so my short answer is I don't think we found like the magic bullet yet.
Very cool. Thank you. Okay, I'd like to invite Adam and Avia back up to have a conversation with you. Core Intent is basically, both of them performed really well on this challenge. And I'd love them to sort of be involved in the design of the next challenge. Yeah, do like take a seat instead of yeah, to take a moment to think through what the virtual cell challenge will look like in 2026, 2027, and what versions of it would yield something akin to an AlphaFold. We are, as a collective, trying to iterate both on the techniques for doing well on the challenge and modeling these systems, but also creating the data sources. And I think you have very different approaches, one having a foundation model style, large-scale training approach. So yeah, I'm just looking forward to hearing also what you're curious to hear from the creator of the challenge and vice versa.
All right, so I was rudely looking at my phone while you were presenting because I was writing some spicy questions. So one question is, the ARC Institute, they released EVO, then they released EVO 2 most recently. Really phenomenal, both models. You didn't talk about it so much, but you've released State. What is State 2 looking like? Is there a State 2?
Hey, thanks for the question. So, so I also think EVO is amazing. I'm not sure why you mentioned EVO before you asked. Do you think you're asking is there a connection between EVO and State, or? So.
What's the sequel?
Sure. Yeah. I mean, I think so. I can respond to this in the context of State independently. I think there's during the course of this talk, probably you also sense for me that I don't think we've reached like virtual cell. I think we have a long way to go. But I think it's really exciting because as we're kind of working towards this, as some of the previous talks also talked about, we're kind of learning where like the pain points are. And I think what we are working on a State 2, if you want to call it that. And we are, there are a lot of things we want to make better and already have better, sort of improved upon internally. So, things like what really is kind of that core biological signal that we should be predicting, right? Is it really meaningful to be predicting everything that you see in a Perturb-seq experiment, or is there something else that is like actually reproducible? So there's a lot of these kind of more interesting questions that we're diving into deeper. We're also going to include a lot of ARC data. So far we've not released any ARC data. In fact, State wasn't even meant to be kind of, well, we kind of released it ahead of schedule. Like the plan was to train a model on ARC data, but because we had some ideas, we released it earlier. So there's going to be some of that. And yeah, we're going to invest a lot of time in VCC too as well. And so we're already kind of putting together all the feedback we've gotten from the community, Yeah, I mean, especially love to hear from the VCC contestants and how it worked for them, and really, I just hope it was a positive experience working on this challenge. You know, like I hope it wasn't boring. So.
Yeah, I guess a couple follow-ups. How do you think about better metrics if we're restricting ourselves just to perturbation prediction performance, in getting at metrics that are hard to hack and maybe push towards more of the sorts of questions research biologists care about in terms of explainability? Like, for instance, have you considered gene regulatory network prediction, things like this?
Yeah, thank you. No, I really appreciate it actually how you brought that up in your talk as well. The importance of explainability, I definitely agree, it's really critical. We have begun to explore gene regulatory networks a bit more deeply. My concern usually with gene regulatory networks is that even the ground truth networks just have so much noise associated with them. I think even when we did GEARS almost like 3-4 years ago now, most of the papers in the field were comparing against GRNs that themselves had, maybe 2-3% precision on. So our feeling was, let's plan to move the metrics a little bit away from just GRNs. But I see why why it's really important for sure, like especially understanding causal structure. So the short answer is yes.
Kind of building on that point, Adam, about the benchmarks. The question for maybe both of you is, Adam, you have a company. You are part of a company or an institute. Recently, I read the newsletter from the Virtual Cell Challenge that companies are asking to license state. And so in this idea of, okay, sure, there's a good benchmark of can we do gene expression? Well, we could have a benchmark of when are we hitting a virtual cell model? But let's think about the benchmark of commercializability. What do you think are those commercializable benchmarks that you're getting asked about with your models? How are people trying to commercialize it? Or how are people trying to use it right now for their businesses?
So, I mean, we have, I guess we have probably lesser experience than maybe Adam or other startups do. In our case, we just essentially are licensing out the use of state. So it's like a pretty standard license. We're seeing more interest from pharma companies and some small biotechs. But this is definitely not been an area of focus for us. We've been much more focused on the science. That's kind of, I guess, one of the advantages of being at a research institute. We're not that driven by showing commercial success, even in the near term. We're okay with waiting until we have something cool, or so we're focused much more on like, can we do scientific discovery? But I'm curious to hear.
Yeah, it's unclear what the assuming we get this GPT-1 moment or AlphaFold 1 moment of virtual cells, what the initial commercial applications will be. I think people have some ideas beyond, like, we're going to use this to replace wet lab experiments. And I alluded to some of them in our talk, like target validation, mechanism of action, elucidation, maybe biomarker selection, stratifying patients. I think my take is that until these models are better and we scale them up, another few orders of magnitude, the really killer apps are not going to be apparent. It's pretty hard to forecast right now, like what they will be used for. Maybe it'll be something completely beyond pharma, for instance. But it's the early days. I guess the follow-up question is, how do you think about, like, what is the threshold for the GPT-1 moment of virtual cells? Is it, getting within some delta of experimental reproducibility on these metrics? Is it like having a killer use case?
Yeah, no, that's a really good question. When folks ask me this, I think the biggest indicator would be like biologists actually using these tools like regularly and meaningfully. Right now, it's more just like a curiosity that you're like, oh, like, what does, let's make a UMAP or whatever, and like actually not use it very much. So I think really kind of becoming part of the standard workflow, like whatever that takes. I mean, I like to give this example, like ChatGPT, for instance, right? Like, it wasn't perfect when it first came out. It was, it had a lot of like hallucinated responses. It was, getting a lot of things, really obvious things wrong, but it was still super useful that people were already using it for so many things. And I think that's kind of the way I look at virtual cells too, is I think people have these really high-flying notions of what they are looking for, whether they want to like explain all of the cells function, but I think even if you can do a reasonable amount of that, such that scientists are using the tool and they're improving like the efficiency of experiments, I think that to me is already a big sign of success.
All right, so we both talked about AlphaFold, and a question related to that, and for both of you actually. So, would you consider AlphaFold a foundation model? It's very specific to structural prediction, but it has a lot of other tasks that it can do. And how do you kind of recognize when to use a foundation model versus when to not? And yeah, how do you identify that difference?
Yeah, short answer, no, I wouldn't say AlphaFold is a foundation model. So why do I say that? Of course, the criteria, you have a lot of domain-specific inductive biases. You're still relying on MSAs, like essentially evolutionary lookup tables, but done in ML kind of way. I don't know if they still use the point invariant attention. I'm not sure what the most recent architecture is. Like certain symmetries built with the model, and it's, it doesn't really generalize beyond this particular task of going from sequence to structure. Maybe the inverse problem. There are ways you can use this as a base. Why is this? It's because we don't have a lot of structure data. But like, if we had more structure data, you could probably do a really scaling, general model, low inductive bias approach. But I don't foresee that becoming possible unless we start generating some amounts of molecular dynamics data. For all these reasons, I think single cell is a lot more amenable to scaling up because, like as Yusuf said, 500 million plus cell profiles already, billions in the coming years. We have an abundance of data. Seems pretty right for scaling.
Yeah, sorry, what was the exact question?
Is AlphaFold a foundation model? How do you decide when to use a foundation model versus not using a foundation model?
Oh, right, yeah. Yeah, so AlphaFold, by the definition of the foundation model, you're right, is not foundation model. Probably a better example of a more foundation model is like ESM, perhaps. Yeah, how do you decide? I guess the simple litmus test is, does pre-training help your task or not? And I think for single cell tasks, we definitely see a benefit of pre-training. In other cases, it's a bit more mixed. But I am definitely optimistic that there's a lot of potential here. I think the other really cool thing, honestly, to get people psyched about single cell is, we are like definitely generating a lot of data and we have a lot of ability to generate it. Like I think if you think about protein sequences and if you think about even DNA, there's definitely a little bit of a bar in terms of how much you can sequence and how much additional data you can get, right? There's only so many proteins in the natural world that I think you can like meaningfully sequence. But I think, but I think with single cells, you can actually generate cell states in like very wide number of conditions and across different contexts, cell types, cell lines. So there's a lot of potential here, definitely in terms of like longer term. So I definitely share the optimism that we can do a lot. But I always say this, that cells are really messy and they are not the same as proteins. So it will also take a lot of careful both model design as well as data set design ideas to like really kind of get the best out of this. But look forward to seeing what that. I actually had a question for you guys too. So I'm always very curious to hear what like the general experience has been for you working on this virtual cell challenge. Has it been like where there's been other like single cell perturbation cell challenges before. Did this challenge was it like more exciting, more interesting? What could have been better? What should we focus on next year? And like my previous question, I hope it wasn't boring. Was it boring at some point? Did it get boring after a long time?
Yeah, positives. I think the data quality is extremely top notch. From the diversity of the perturbations, the sequencing depth per cell, like median 50,000 UMI. For context, this is how many RNAs basically are you drawing per cell, and public data sets range from anywhere from like 1,000 to maybe like tens of thousands on the higher end, and they are put out this amazing high quality, high sequencing depth data set. I'd say maybe some small critiques are the metrics feel a bit arbitrary in some ways. And like someone asked about the absolute value operation and the PDS metric. I think the DE metric's great. idling with the Wilcoxon rank sum test size. Lots of options you could choose there. Yeah, going forward, maybe just keep up the high quality data. It's really excellent.
Yeah, heard the name Virtual Cell Challenge. It has been a challenge. It's been, and the thing is, I started this out not knowing much about single cell, and I haven't been doing that many machine learning projects recently as an IC, so I have to really up my level there. And I told myself also that I would stop when I stopped learning, and I haven't stopped yet. So every day there's always like an aha moment. Oh yeah. And so I think this is thinking about this challenge, as I said before, is this, maybe the outcome that we're looking for is not an AlphaFold, at least in the first year. But in the first year, it's like getting people excited, getting people learning about this problem and trying to figure that out. Specific details, Adam mentioned metrics, so I won't talk about that some more. I think the data piece is very interesting. The fact that we can leverage data from all different locations. I do worry, as a person who doesn't have a lab, whether like how easy it would be for a lab to just generate perturb-seq on H1 ESC data, and that being the ultimate success. Still probably a win for the Virtual Cell Challenge, but may not be the most interesting answer to the problem. And so, yeah, maybe perhaps thinking about how to restrict or how to expand the amount of data that's being used. And then kind of bringing it back to a question to you is, this year, you focus a lot on context prediction. What would subsequent years look like?
Well, thanks. It was really great feedback. I think the perfect response would be exactly what you said, like that you're learning something interesting every day. That's really the goal for this, is to get people excited about this problem. It's too early for me to say decisively what next year's challenge is going to be. Rest assured that everyone's feedback is being taken seriously. All the Twitter rage threads are being accounted for. All the private comments are like through the Slack, what do you call it? The Discord. All of that is we're definitely accounting for all of that. For me personally, I think if we stick to the context generalization setting, I would definitely like it to do multiple contexts in the same data set, so you don't have to deal with the annoyance that is training on Global or other data sets that are different modality, different sequencing, different CRISPR sort of setup. So we will try to make sure that's available. At ARC, we've also been doing a lot of stuff with in vivo perturb-seq, so that's something that's definitely very interesting. Maybe too early to say for sure, because I just dropped this right now, but like it would be really cool if we could include that as part of the competition. But yeah, besides that, we're open to other feedback. We're definitely going to be staying in genetic perturbation space, and not sure if we'll be adding additional modalities just yet. But it seems like there's a lot to be done, even in single images transcriptomics. So.
That makes me think of one other kind of feedback I thought. That is, and it's related to the point that Adam made. Is, we have benchmarks, and the goal right now is gene expression prediction. But also, again, the point about commercialization. Perhaps I wonder if there's a very relevant benchmark that is useful for helping patients, or that is useful for growing a business, or that is useful for solving a scientific problem directly. Now, I do think that the benchmarks right now are really elegant in the sense of I found myself optimizing for PDS, and then my DES score was really poor. And then I optimized for the DES, and then my DES score went really poor. And so, in a way, there's this nice hug and push, pull and push that's helping me make my model even better. But I also wonder whether having benchmarks that are very application pragmatic specific may be an interesting feature for the virtual session.
Well, I was gonna say that's definitely very important, and I think we just, I guess, have to be careful to make sure there's enough of a dynamic range on those benchmarks too, and so I think, yeah, so that's, but I agree, I think one of our goals was to keep them complementary and to have that struggle between DE score and, because I know that you can hack the DE score by doing the average of over certain cells, and, but then the idea was that then you would hopefully do worse on perturbation discrimination, but I feel like people have also hacked that now, and I don't know what's going on. But it's going to be exciting, I think, once we get the final results. Yeah.
Yeah, I guess to finish up, you guys have any suggestions for people who are about to set up their infra for the first submissions?
Use the proper tokenizer. There's some weird stuff about dealing with the sub-G matrix. Be wary of zeros. You're going to encounter a lot of zeros. Think about how to process those. Look at the metadata, stratify by the metadata.
Yeah, I mentioned a few pieces of advice. Again, I would dive deep into self-eval. That's very key. And just really understanding how that works and how they're evaluating. Because you can do everything else right, but then if you kind of mess up the last step, then you won't be learning. You won't be getting the score and the feedback that is right. So yeah, really deeply understand self-eval, how that works. And then for those, before the submission process, think about your team. Think about the areas that you want to add on to the knowledge pieces that you're missing and you want to add on. So if you are very tech focused, then think about adding some biologists. If you're very biology focused, definitely think about adding some of the tech people.
Yusuf, any thoughts?
For the competition, yeah, I mean, I think that covers most of it. We, unfortunately, we were not allowed to take part in competitions that haven't actually even gone through, tried the interface, as someone who was behind the scenes and like looked at all the data, including the test data, yeah, I think those sound like the right steps. And it's also useful to look through the blog that describes how we created the data set. There's a certain distribution across the perturbations you're looking at. Some of them are very strong, others are very weak. Metrics often behave very differently on different strengths of perturbation. So yeah, just kind of carefully looking through what you're predicting and why you're like Rio said, like thinking about why the metric changes. I think those subtle things will give you a lot of intuition. Also, Abhi's put out some nice notebooks. I know they're heavy, but like I think just walking through that process will give you a feel for how you could build your own model that doesn't have to be safe, but you know, could just kind of reuse some of those data loaders or evals, or you know, just kind of get you up and running more quickly.
Fantastic. Thank you. Okay, and with that, let's get to building our own virtual cells. So this is the moment of the event where I hand the mic off to you. You raise your hand real quick if you'd like to pitch your team or your idea for approaching the problem. It's a basically 60-second pitch to attract a teammate, if you're curious. So, okay. So if you're interested in pitching, that's fine. You can use the Slack channel to coordinate, which I'm about to send out. You guys can sit down. Yeah, I guess quickly, the plan for the afternoon and evening will be that we'll train virtual cell models for the next 7 hours. So the demos will be at 9. We'll be at 9. We edited talks a little bit later, so we'll have a extension on the time so that people can get to submissions. And then the first, second, and third place will be as evaluated by the challenge. And hackathon winner, as well as folk who do well overall. We did have a snafu with lunch, so lunch was supposed to come about 30 minutes ago. It partially arrived, so we reordered it, and it'll be here in about 40 minutes. So there's a little bit of food, but not a ton of food. A ton of food is going to show up in about 40 minutes, so do keep an eye out for it. yeah, I guess high-level logistics. So you're on the 3rd floor of a 4-story mansion. Please do not go upstairs, but feel free to use this floor, the floor below, and the floor below that. This bathroom may or may not be out of service, so if it's not working, there's a bathroom 2 floors below this one that is functional. Okay. Okay, and with that, good luck. Let the hackathon begin. So Hyperbolic was able to give GPU access to those who signed up on Thursday. If you weren't able to get access, do try to team up with someone who does have access. They have $100 in GPU credits for Hyperbolic. If you weren't able to sign up in time, I did send a request for them, and I'll see if they can accommodate a same-day request. But the form did close out here.
He's on the way. No, I mean,